Good morning class, welcome back to microbiology. This is Silvana O'Coney and today's lecture, lecture 2, is about prokaryotic cell structure and function. The cytoplasmic matrix, or the cytoplasm, is simply the substance in which the nucleoid, ribosomes and inclusion bodies are suspended. Prokaryotic cells lack organelles, and of course organelles are structures bound by unit membranes. Prokaryotic cells are composed largely of water. In fact, about 70% of bacterial mass is water. The cytoplasmic matrix is a major part of the protoplasm, that is, the plasma membrane and everything within. For quite a long time, it was thought that prokaryotes lacked cytoskeletons. But recently, homologs of all three eukaryotic cytoskeleton elements have been discovered. And you might remember these three major elements from living systems. They're called microtubules, microfilaments, and intermediate filaments. These images here show the helical arrangement of the cytoskeleton within cells of the bacteria Bacillus subtilis, a very common soil bacterium. And these cytoskeleton elements carry out similar functions to those in eukaryotic cells. Many prokaryotic cells also contain structures called inclusion bodies. Inclusion bodies are granules of organic or inorganic material that are stockpiled by the cell for future use. Some inclusion bodies lie free in the cytoplasm, others are enclosed in a shell, which is a single layer composed of proteins, or it might be a membrane-like structure composed of proteins and lipids. Inclusion bodies are mostly used for storage, but they also function to reduce osmotic pressure within the cell by tying up certain molecules in particulate form. Let's now look at some examples of inclusion bodies, starting with organic inclusion bodies. The major type of organic inclusion body is the glycogen body. Glycogen, of course, is a polymer of glucose units composed of long chains, and it's also found as a storage form of glucose in animals, where it's stored mostly in the liver and muscle cells. Now, just like glycogen inclusion bodies, PHBs, polybeta hydroxybutyrate inclusion bodies, are carbon storage reservoirs, and these provide material for energy and biosynthesis. Many bacteria can also store carbon as lipid droplets. The photosynthetic bacteria, otherwise known as cyanobacteria, have two distinctive organic inclusion bodies. The first type are referred to as cyanophysin granules, and these are composed of large polypeptides. They function to store excess nitrogen for use by the bacteria. The second type of distinctive inclusion body found in cyanobacteria are the carboxysomes. These inclusion bodies contain the enzyme rubisco, which is a critical enzyme for the fixation of carbon dioxide. And carboxysomes serve as a reserve for this enzyme. Another example is the gas vacuole. This is quite a unique inclusion body which provides buoyancy to aquatic prokaryotes. Gas vacuoles are aggregates made of a single type of protein, and this protein is impermeable to water but highly permeable to atmospheric gases. So these bacteria can regulate buoyancy to float at the appropriate depth for proper light intensity, oxygen concentration, and nutrient levels. They sink by collapsing the vesicles, and they float upwards when new gas vacuoles are constructed. There are two major types of inorganic inclusion bodies seen in lots of bacteria. Polyphosphate granules, for example, are used for the storage of phosphate. Polyphosphate is simply a linear polymer of phosphates joined together by ester bonds. Polyphosphate functions as an important component of cell constituents, such as nucleic acids, and polyphosphate can also serve as an energy source in cell reactions. Sulfur granules are also inorganic inclusion bodies, and these are used by some bacteria for the temporary storage of sulfur. The cytoplasm of a prokaryotic cell is usually packed with ribosomes, and some ribosomes may also be attached loosely to the plasma membrane. Ribosomes are complex structures consisting of protein and ribosomal RNA, and you might recall that ribosomes are the protein factories of the cell. Prokaryotic ribosomes are smaller than eukaryotic ribosomes. They're called, prokaryotic ribosomes are called 70S ribosomes, while eukaryotic ribosomes are called 80S ribosomes, where the S stands for Svedberg unit. This is refers to the sedimentation coefficient, which is a measure of the rate of sedimentation velocity in a centrifuge. So the faster a particle travels, the greater the sedimentation coefficient, or S value. The heavier or larger particles normally have larger S units. The most striking difference between prokaryotic and eukaryotic cells is the way the genetic material is packaged. As you know, eukaryotic cells have 2 or more chromosomes contained within a membrane-bound nucleus. Prokaryotic cells lack this, although there are some exceptions known where the bacterial chromosome is surrounded by a single or double membrane. Prokaryotes usually only have one single chromosome, but some do have a linear DNA chromosome rather than a circular chromosome, and some do have more than one chromosome. So prokaryotic chromosomes are located not in a nucleus, but in a discrete region in the cytoplasm called the nucleoid. You can see in figure a bacterial cell that's actively dividing into 2 cells, and the chromosome material has been stained specifically with a red dye. In figure B, we can see an E. coli cell, and the chromosome has been released from this E. coli cell. The chromosome of E. coli is about 700 times longer than the cell, so that means it must be very efficiently packaged to fit within the nucleoid region. The chromosome is looped and coiled extensively by the aid of condensing proteins, which are very highly conserved in prokaryotic cells. These proteins are not like the histone proteins found associated with eukaryotic chromosomes. So in summary, the prokaryotic chromosome is a closed, circular, double-stranded DNA molecule. Well, in most cases anyway. The chromosome is looped and coiled extensively to enable it to be packaged within the nucleoid region. This packaging and condensing is probably assisted by specialised nucleoid proteins, and these proteins differ from the histone proteins found associated with eukaryotic DNA. In addition to the chromosome, many prokaryotes and even some yeasts and other fungi contain additional DNA molecules, extra chromosomal DNA molecules called plasmids. Plasmids are usually small, closed, circular DNA molecules, and you can see some plasmid DNA in this image here. Although this is certainly not to scale, plasmid DNA molecules are generally much smaller than the bacterial chromosome. Plasmids are usually circular, but some linear plasmids have been identified. Plasmids have relatively few genes present, normally less than 30 genes. They're able to replicate autonomously. There are single copy plasmids which produce only one copy per cell, and multi-copy plasmids, which are generally smaller, that can produce 40 or more plasmids per cell. Plasmids are inherited stably during cell division, but they're not always equally apportioned into the daughter cells. So, for example, one daughter cell might receive 2 plasmids, and the other daughter cell might receive 30 plasmids. Now, an important point here is that the genes found on plasmids are not essential to survival of the host, but they may confer some type of selective advantage to the bacterium, for example, drug resistance. And we'll see a few additional examples in another slide. Sometimes plasmids can be lost from the bacterial cell. This process is called curing. So, for example, plasmids could be lost as a result of UV and ionizing radiation, growth of the bacteria at excessively high temperatures, thymine starvation, and the presence of antibiotics. Plasmids are classified based on their mode of existence, spread, and function. Here we have a summary of the major types of plasmids. Conjugating plasmids are one type. They contain the genes which encode the construction of hair-like structures on the outside of the bacterial cell. And these structures are called pili. Pili can transfer copies of those plasmids to other bacteria. The fertility factor, or F factor, which you can see at the top of the list here, this particular plasmid or factor was identified first in E. coli, and it's the most widely studied of all the plasmid types. The second type of plasmid that you can see listed here is the R plasmid, also called R factor, where the R stands for resistance. And R factors confer antibiotic resistance on the bacterial cells that contain them. Typically, R factors contain genes that code for enzymes that can destroy or modify antibiotics in their immediate environment. R factors may have a single resistance gene or as many as 8 antibiotic resistance genes. These resistance genes are often found within mobile genetic elements called transposons. You might remember transposons from living systems. So it's possible for multiple drug-resistant plasmids to evolve. This is quite troubling because these can rapidly spread through a population of cells, and some are even readily transferred between different species of bacteria. The next type of plasmid on the list are the col plasmids, C-O-L, and these plasmids produce proteins that destroy other bacteria, usually closely related strains. So, for example, E. coli produce colicin bacteriocins, which are directed against other E. coli bacteria, and this may give the bacterial cell a competitive advantage. These bacteriocins kill other cells by forming channels in the plasma membrane. Next, we have the virulence plasmids, and these plasmids encode factors that make the host bacterial cell more pathogenic. So, for example, it enables the bacteria to produce toxins and substances required for the induction of tumours. Finally, we have metabolic type plasmids, and these plasmids carry genes for enzymes that can degrade certain substances. So, for example, some of these genes can degrade aromatic compounds such as toluene. There are others that have been found to degrade pesticides such as 2,4-D. Others can degrade or metabolize sugar such as lactose. And also there are metabolic plasmids that are used for nitrogen fixation by rhizobium bacteria.